Hi, everybody, and welcome to another special episode of the Combinate Podcast. I'm your host, Subi Sadeh. On this episode, I was joined by my good friend, David Simeons, an expert in lyophilization. In this episode, David discusses the lyo freeze drying process, focusing on the environmental and equipment considerations necessary for a successful lyophilization process. This is a continuation of an episode that we did a few weeks back. That one was focused on how the lyophilization process works. This one is focused on what makes it work. In other words, the equipment that makes it all happen. So in this episode, David walks us through the lyophilization process from the point of view of the equipment, talking about the critical subsystems that are required, like I said, to make sure that the process is successful. He also talks about the evolving automation trends in lyo. He talks about special considerations for handling potent drugs and non-aqueous solutions, instrumentation, and a whole lot more. I hope you enjoyed this episode with David. If you're not already subscribed to the show, please subscribe on whatever platform you're listening on and give it 5 stars. Without further ado, here's my friend David.
Hello, Combi Nation. My name is Subi Sadeh. I've spent over a decade in medical device, pharma, and combination product development. Our industry feels complicated sometimes. Drugs, devices, clinical trials, submissions, sterilization, validation, design control, risk management, market access, reimbursement. The list goes on. My goal is mastery, so this podcast is to ask questions I have to people who may have the answers. Each week on the Combinate Podcast, I talk to someone about their area to further understand and simplify. Whether you're a pharma person trying to understand the next wave of products, or a device person trying to navigate a pharma system you're unfamiliar with, or a newbie in both areas, I invite you to listen, and together we can simplify by combinating.
I would say if you haven't listened to the last episode, I would maybe encourage you to start there. In that one, we talked about freezing, sublimation, desorption, and backfilling, and then the stoppering of the vial, explaining the stages that the vial goes through until you have a beautifully filled lyo vial with an excellent cake. In this episode, we're going to talk about the environment and the equipment that make all of that happen. So, David, maybe explain to us the filling environment, the environment inside the freeze dryer, and then what considerations have to be made for the fact that in certain instance it's not fill, stop, or done like a
Yeah.
serum situation.
Yes. Yeah. Freeze dryers can be used in a number of different industries with a number of different products. They're used even within food or within bulk processing. The environment I'm most familiar with or I work within is probably the most stringent, which would be in an aseptic sterile type products. What I've been speaking to has mostly been in like grade A, ISO 5 airflow spaces. So either under an isolator or whatever the case is, they're being filled under that grade A air and then also being transferred under that grade A air to the freeze dryer. I think something that sometimes can be missed is these freeze dryers in a commercial setting, especially when you're having to maintain a sterile aseptic field, these freeze dryers go under a number of different sub processes that are supported by the freeze dryer and the subsystems. So the freeze dryers have a bunch of different subsystems that support it. And by utilizing these in different ways, you can create a clean environment and a sterile environment. So that can usually be done either after a product run or a batch, and then typically before a batch or campaign.
So actually, maybe explain to me, because I don't have a lot of experience with cleaning validations, but is what you're talking about just maintaining the environment?
Yeah, and so there isn't laminar airflow, right, within the freeze dryer. It's creating a sterile environment, just very much like an autoclave would create a sterile environment or sterilized product. Or if you're just running an empty cycle with an autoclave, you would do something similar where you're creating that sterile environment within the freeze dryer prior to introducing the vials or the product and doing the freeze drying cycle. The cleaning validation then would be on the other side of that, right? After you're unloading your batch, that's when cleaning validation would be relevant. And there is a whole separate process to go through that for cleaning validation around a lyo.
Okay. So the filling process during filling, the vials flow, they're filled with liquid, then
Mhm.
they're half stoppered, then
Correct.
they're loaded into the trays that eventually go into the freeze dryer. The freeze dryers are typically attached to the filling like line areas, or is there some like whip area that they go
So
in?
So it goes back to the last podcast, and it really depends on where the facility is at. It goes back to when I said row by row versus a T car or a transfer cart. A lot of that can be automated and is automated, and especially like with Annex 1 in the EU, they're pushing towards automation, removing the operators. Historically, there has been, yes, that kind of whip area where operators are loading trays, moving them to HEPA carts so that they're still within a grade A environment, and then transferred from the HEPA cart to the freeze dryer. That's becoming less and less common, especially with that Annex 1 push of removing the operator, making more things automated. The row by row is directly attached typically to the line, either through different conveyor systems or whatnot, where it goes in and out of the freeze dryer before it goes to cap crimp. Multiple lyos can be used in that case, from using like a T system of conveyors. And then there's also the other system that I was talking about with the T cars on rails, where you can have essentially as many freeze dryers as you want. I've seen them up to 4 freeze dryers, up to 8 freeze dryers, big, large freeze dryers, where different campaigns can be run with the T car delivering it automatically to the freeze dryer.
Hmm. And what is a HEPA cart?
A HEPA cart is exactly what it sounds like. It's a cart that has HEPA filters
Oh,
installed on top of it. So basically, it's a type of a RAB system, I would say, where you can open doors, load it, and then when you close the doors, it's a closed environment with HEPA airflow.
and you fit a lot of vials on it?
Oh, sure, yeah. The larger ones, yes. It could be probably like 5, 6 shelves worth of vials. It In my mind, it's not as practical these days, but it's something that was historically used and might still be used in facilities to date.
Okay. Then double clicking into the lyo chamber itself. How do you make whether it's a lyo or an autoclave or that, the room is prepared before you take the vials in?
Yes, yeah, the
Okay.
Lyo would definitely be prepared. So it would definitely go through, and I think I've talked to you about this previously, but there's some acronyms: SIP, or a CIP, SIP. Stands for clean in place, followed by a steam in place. So the clean in place is removing any kind of particulates, where the steam in place is actually doing the sterilization. All those things would go through qualification validation during your commissioning phases, obviously prior to using it in an actual batch, so that your cycles are validated. But those would be used prior to a batch. And typically there's some form of like leak testing or vacuum integrity testing to make sure that the system is tight, that there's not air getting in. Remember that freeze drying's basic principles are under deep vacuum, low pressures, especially compared to atmosphere. So if you have a leak in a gasket or in a tri clamp, you don't want out outer air coming into the freeze dryer. So that's where that leak test is also essential.
What does that testing look like?
There could be a number of different things to look at that. The equipment has some supporting equipment for that. Leak testing can have like a rise test, where basically you pull a vacuum of some nature and then you're seeing over time how much the rate goes up and whether that would be acceptable.
Walk me through the equipment that's used for lyo.
Yeah, it's just high level. There's the chamber, which is where the product sits, and I'll go into more detail on each one. And then directly attached to it would be a condenser. This is where most of the solvents would end up and refreeze before it's then defrosted at the end of the cycle. But the condenser, so the chamber and condenser are by far the largest pieces in the main two kind of chambers. So the chamber, condenser. And then there's a refrigeration system, um, a vacuum system. Those four components or systems are probably the majority of the freeze dryer. There's also other supporting equipment like a filter rack, as far as being able to filter your air, control your air, um, whether it be air or nitrogen. So those are additional supporting systems. Looking at more detail on them, the chamber again is where the vials sit. So usually vials go through a slot door and are pushed onto the shelves. The shelves are what are within the chamber, and then a hydraulic piston would move the shelf stack up and down automatically, usually trying to meet the height of the slot door. So there's a smooth transition for the vials onto each shelf. The chamber then would seal off with the use of the slot door gaskets prior to starting the freeze drying. So you're preventing contamination from either leaving the freeze dryer or entering the freeze dryer. Each shelf is typically hollow inside, and that allows for the cooling fluid, usually like silicone oil, to flow through the shelf to the refrigeration system. That helps you precisely control your temperature, whether you're going up or down with the shelf temperature. So that's the
The
main
shelves. Okay,
temperature.
so that's interesting because to cool the shelves,
Mhm.
silicone oil flows through them.
Yes, typically, yes. Old freeze dryers, they used to actually put the refrigerant through the shelf, but you could see potential issue there with a leak. Refrigerant obviously is much more difficult to clean, and you wouldn't want that in your freeze dryer even if it was empty. So now most refrigeration systems use some kind of refrigerant and heat transfer to cool or heat the silicone oil, which then is in a loop through the shelves.
Yeah, so there's obviously no expo because I know silicone migration in manufacturing is a huge problem, and so there's no exposure obviously
No,
to the silicone then. Okay.
no, no. This is flowing through stainless steel
Stainless
shelves.
steel. Yeah. Yeah.
Yeah.
Okay. On you go. So is that everything
Yes.
related to the shelves or is there anything else?
I think that's the main parts really to discuss here. I think then leading to the condenser. So how do you control your between the condenser and the chamber? And that's
Sorry,
usually,
before you
yep,
go to the condenser, I guess what is are all of these things are inside the chamber.
everything I've discussed so far.
But I'm saying not just the shelves, but I'm saying the condenser, the vacuum, the refrigerate, the refrigerating element, and the filter rack. They're all physically inside the box of the chamber. No,
The chamber is standing alone. The only thing in the chamber is the shelves and the
it's an
product.
empty room, shelves, product, chamber. That's
Yeah, I
it.
wouldn't look at it as a room, right? Like the equipment itself. So you have your big box, essentially, and this is going to try and simplify it as much as possible. But your big box is your chamber, right? Within the chamber, you have your shelves, which your product sits on, and then you have your coils that are going to inlets and outlets of the shelves, and that's it, really. That those are your main components in
Hmm,
the chamber. Everything is stainless, right? Everything is because you need it to be cleaned with a CIP, SIP.
got it.
And then the condenser would be directly either adjacent to it or under it. So different manufacturers make them different ways, and to end user request, whether it's on the side or whether it's on the bottom. Segregating those two pieces is a main isolation valve. The main isolation valve could be, historically a butterfly valve, a mushroom valve. They're very large, and basically that's what opens and closes between the chamber and the condenser. So now getting into the condenser. So the condenser is also connected to the refrigeration system. It has coils that circulate refrigerant, and that refrigerant enters into a liquid state and exits in a more of a gaseous state after removing the heat from the surrounding environment. The vacuum system's connected to the condenser as well and allows for the pressure to be lowered in the chamber and condenser, and then the gas to move from the chamber to the condenser and then exhaust out. The temperature of the refrigerant coils is set to always be colder than the shelf temperature in the chamber during the drying processes, and this is essential to allow the solvent to condense and freeze onto the coils.
So if I understand it correctly, right, looking at it as an empty room is the incorrect way to look at it.
It's a piece of equipment.
It's a piece of equipment with a door.
Mhm.
You open the door there, unless it's automatically loaded, but presumably you open a door, vials are loaded in onto shelves. That's what behind all of that, behind all of that, there's a condenser and then a vacuum. The vacuum sucks from the inside of the chamber through the condenser into the vacuum. And as the vacuum is pulled, and as you get closer to that freezing temperature, and the vacuum is pulled near complete vacuum, we went through the triple point of water in the last episode. That solvent is like sucked out of the sucked out of the chamber into the condenser, and the vacuum is maintaining that pressure differential. Okay, makes sense. I guess I had it thought about it in I had it in the way that I envisioned it was not that it's like this parallel system that just goes from one side to
Yeah, I
the other.
mean, a very easy comparison, and no, they're not the same thing, is an autoclave, right? An autoclave is a piece of equipment. It's a box. It has shelves that your load goes in. It has the sorting equipment to make the process. Same thing with the freeze dryer. You have your chamber, which is your box. Your product goes in it, and then you have the supporting equipment.
Yeah. The refrigerator.
Yep, I can go into some details there.
Yeah, let's do that.
So for a refrigeration system, this system heats and cools the surfaces within the chamber and the condenser. It contains
So it's called a refrigerator, but it's basically temperature control. It
so
heats and cools.
refrigeration, yeah, refrigeration system, but the refrigeration system typically has a refrigeration component to it and also a heating component to it.
Okay. Is it called a refrigerator because of freeze drying, it's typically colder than ambient?
I would say it's called a refrigeration system because the vast majority of the components on that system are for the purpose of creating extreme cold temperatures, right? And you're using refrigerant to do that. The heating aspect of it is usually there are three heaters that are part of the system that also act to heat the loops. But the vast majority of the refrigeration system or skid is for the purpose of refrigeration, for
Mm,
cooling. For example, three compressors on the refrigeration system, and then silicone oil circuit that's circulated with a pump. It has two purposes. So you're cooling the silicone oil down through the heat exchanger between them, and then directly cool the condenser coils and a jacket around the chamber. So the chamber itself has a jacket, very similar to a formulation tank having a jacket. So that's also used to cool, running the refrigerant through it, and then or heat itself by running through the silicone oil heater.
I was gonna
So
say I'm assuming their jacket isn't as sweet as your velvet jacket.
yeah,
Ah,
I like it. So
yeah.
so keep in mind what we're trying to achieve here is temperature control of the shelves, the chamber, and the condenser coils.
Is there any equipment we haven't gone through?
The vacuum system.
The vacuum.
Yep. So the vacuum system, so that's what draws the vacuum down from the condenser. So the vacuum's drawn down in the chamber when that main isolation valve is opened
Mm,
between the two vessels. And that includes usually like two dry vacuum pumps, sometimes a booster pump, and then that will usually exhaust through a HEPA filter to collect any residual particulate, especially like in a potent manner. Sometimes if you're not doing potent or solvent or hormone type products, you don't necessarily need that HEPA filter, but it is definitely used in those cases. So the vacuum system, it's the point is vacuum pressure control of the chamber and the condenser, which is essential for effectively evacuating solvent and keeping high quality freeze drying performance.
mm. The filter rack.
Yep. So the filter rack I touched on, to keep it simple, it's used to provide point of use clean air, clean nitrogen. There are some instrumentation we didn't talk to, which would be like pressure gauges, right? Pirani gauges, capacitance gauges or instrumentation. So those would be reading, say, your actual pressure versus your pressure with solvent or water, right? Those are instruments that could be used to tell when the end of primary drying is happening. So there's definitely more instrumentation.
I was going to say, with a lot of processes, there are consumables that are used. Are there any that are used with?
Not that are coming directly to mind, not at least in a automated commercial type world, right? There's no, sure, you might have to change your filters occasionally, but it's nothing that's like on a batch to batch basis, right?
In
Most of it is automated type systems that support it.
in the last
Um,
episode, we talked about nitrogen sparging. What about, is there any component that's added in during lyo other than the gas?
no physical materials.
Okay. And so it's just filters and nitrogen or whatever gas you're
For
filling that
aeration,
space.
for the backfilling of the vial headspace, yes.
Yeah,
Yep.
that's it.
Yeah, you're filtering in your clean air or your nitrogen. But there's no, if you're talking about filling a vial, you have vials, you have stoppers or cap crimp, you have your caps, like from a material or commodity standpoint. From a lyo standpoint, it's processed. There's no materials or commodities being introduced.
Mm. What are the, like, okay, so we just went through the equipment. What are typical problems that you see when you're qualifying a room or validating a process?
I think typically it's just a lack of understanding the process, a lack of understanding what critical quality attributes are, critical process parameters, the actual understanding of why you're freeze drying. And this can go across the whole sterile aseptic fill finish process, but especially with freeze drying, right? In a world where we are trying to use integrated commissioning, qualification, validation, there's this mentality still of having to test everything or to validate everything where that's not necessary. Like you talk about, um, one of the commissioning tests is you basically fill up huge trays on every shelf and you're trying to test the capacity of the condenser. The capacity of the condenser is not directly related to your critical quality attributes for your product, right? That's more of a how big of a batch can I do or something like that. But I've seen companies that try to still validate their condenser capacity. So there's certain things like that where it's just overkill because I think there's a lack of understanding of what are the actual CQAs and CPPs.
Yeah, this just came to mind. There are, for some sterilization processes, as part of the kind of ongoing manufacturing, a a device or a vial with a biological or chemical indicator will be put
Bioburden, yeah.
by, they'll be put in to ensure that the,
BI, biological
yeah,
indicators.
BICI,
Yep,
exactly.
yep.
Is there anything that's used to, like, okay, this is good enough lyo?
Well, you definitely would be doing biological indicators for your steam in place validation that would take place before any kind of PPQ
Well,
back.
where I was drawing the parallel is not on the sterile fill, but like,
A
I don't know,
product.
like a, like some kind of representative reference sample that would say, okay, we
Mhm.
got the appropriate level of moisture out.
So there's a lot of really exciting technology that's either new from a lyophilization technology standpoint or that's coming. I'm aware of some instrumentation out there that does temperature monitoring live. The tricky part about freeze dryers is they're these huge stainless steel tanks, right? So having some kind of like transponder that is going to be able to communicate live or directly inside the freeze dryer can sometimes be difficult. That being said, there is that technology that's coming or is potentially already out there. With freeze drying too, though, there's also this: the more information we have isn't necessarily a good thing, right? Because what do you do with it? How important is it? Versus like just understanding temperature, pressure, time, and what it means for your product.
I think where I was going is that for autoclaves, there's like parametric release. For gamma, there's like dosimetric release. Is the same thing the litmus test for a good or bad? You execute a cycle. you take out vials that are ready for crimping.
Yep.
And so how do you know that the moisture content of the cake is a success or is a
Yep.
failure?
Yeah, it would go through your process validation where you're running batches and you're pulling your samples to do your validation, analytical testing, quality controls, that type of stuff. They're typically being pulled from the capper after they're sealed, just so that they've gone through the process. You can analyze the moisture and stuff after it's already capped, crimped. And then also during your actual batches, you would be pulling potential samples to do those additional testing or release testing. But as far as there has been some conversations, and I do believe some companies either talk about it or have done it where they introduced, call it product monitoring vials throughout their shelf stack. One of the big challenges in freeze drying is shelf or is vial to vial uniformity across, say, a shelf stack or different freeze dryers. How do you validate or how do you qualify the freeze dryer within the design space to understand that a vial on the top shelf in the back right corner is being freeze dried the same way as a vial on the bottom shelf in the front left corner, right? How can you say that those products are going through the same process? So that's definitely part of the qualification, process validation, design space conversations. But where I was going is some companies are now trying to introduce these basically like monitors that sit inside a stopper that would then sit within a product vial on given spaces of different shelves to say we've monitored the process throughout the whole freeze drying cycle. The
Hmm.
problem with a lot of those types of technologies, and I'm sure some of these things are being worked out, is if you're introducing some kind of probe sits in your product to monitor it. If you think about the principles of freeze drying, take freezing, for example, the liquid is going to freeze differently now that you have something sitting in the middle of it, right? If it's going to freeze differently than if nothing was sitting in it, right? So it's that argument of, is this giving us representative data and information, or is it miscuing how nucleation is actually taking place?
Yeah, in other words, is the introduction of this monitoring device changing how the product performs,
Right.
and is it
Is it
actually presented?
misrepresenting?
Is it
Right.
yeah. Is it creating a different product than the one that it is
Right.
actually being produced? To just like the starting where we began about the intent of Lyo, you had said in the last episode that lyophilization is a stabilization process. And I thought that was I hadn't heard that before, that like it's obvious and inherent, but the intent of lyophilization is stabilization by way of removing moisture. And is the end goal of a lyo cycle to be as close to zero moisture as possible?
It really depends on the product, right?
And I
But
think the reason that I
most of
bring
the
up
time, yes. Yeah.
the reason that I bring up this point about stabilization is about what you were saying about uniformity. Like I'm trying to, and maybe this is the wrong parallel to draw, going to sterilization, where I have a little bit more experience. When you're looking in a sterilization chamber, you put in the BIs and CIs in the areas that you think are going to be the least
Highest risk.
like highest risk, right? And so I'm
You're
wondering,
draining.
when you're talking about the uniformity, can you go too far with lyophilization such that you wouldn't be looking for the highest risk areas? You know what I
Yeah,
mean?
I mean, moisture contents can be like as stringent as needs to be between 2 and 3%, or it could be like less than 5%. So it's all how the product is designed and what you're trying to achieve. That's usually done more on that S&T R&D side of things, more so than what I'm seeing. It's more so, how do you create the equipment design space to achieve it?
So we went through a standard process overview with process and equipment. What are the edge cases and the situations that are less common but still important to consider?
Yeah, I think a big push in the industry right now is, how do you process potent materials with non-aqueous solvents? So, like alcohols that are being used as solvents, hormones, right? So, there's a lot of new drugs that are coming down pipelines of different companies. And process these still in that sterile aseptic environment, and also at the same time protect your operators in your facility. So potent drugs, they can have very low operational exposure limits, or OELs. So there's special considerations in process development and the machine design to make sure that they can be handled safely. Examples would be using critical and non-critical drains, having drain selectability built into recipe capability so that when you're defrosting a condenser, does your first rinse of defrost or your liquid ring pump that's draining water go to critical drain, and then your following rinses go to a non-critical drain. So also from like a sustainability standpoint, leak testing would be essential. So prior to starting your new freeze drying batch, the leak test must be successfully ran to show that you have a sealed system for both a product quality standpoint and from operator safety. Also using potent product interlocks. So following a freeze drying cycle with drug product, your control system requiring an operator to run a CIP cycle prior to allowing any other manual or automatic operation to take place. Because inside the lyo is considered to be contaminated with potent drug products. So you want to make sure you're using that CIP before any further action can take place. Sometimes you can have an override, but that kind of gets more complex. And then from that solvent, that non-aqueous solvent handling standpoint, when a solvent's being removed from the drug product, that's more like an alcohol, these things can be a safety risk for being explosive. And therefore, additional design considerations need to exist. So something like using an initial evacuation with a liquid ring pump that can compress it within a dry environment, or sorry, if it's compressed in a dry environment, it can be explosive. So for that reason, you want to be able to handle that with the liquid ring pump versus pulling that into your dry vacuum pumps.
Mm.
Also, like same thing with interlocks after a freeze drying cycle, using a non-aqueous solvent, the control system requiring an operator to run a condenser defrost prior to any kind of manual or automatic operation, being able to control your Pirani gauges can get hot. So
What is
having
that? Pirani gauge?
Pirani gauges are used for measuring the pressure while you're running product. So it's compared against your capacitance manometer. So you're usually controlling with your capacitance, but observing the Pirani against it. But Piranis can get hot. So how do you make sure that's interlocked when you're using a solvent so that you're not creating an ignition source? So those kind of things are important to consider when designing and or automating freeze dryers.
David, I think that was pretty comprehensive as
It's a
far
lot.
as yeah,
Yes.
that last part at the end, my brain hurt a little bit, but where can people find you, David?
Yeah, you can find me on LinkedIn, either David Simeons or Life Science Professionals. I have a company page on LinkedIn. It's pretty bare right now as I just went out on my own last year, and then a website hopefully dropping here soon. Otherwise, just David at lifescienceprofessionals.com.
Thanks for coming out again, David.
Yeah, thanks again, Subi. I look forward to the next one.
Yeah, all right. Looking forward to it.